Welcome to the 59th session of Beyond Research. I am Swapnita Das and I'll be your host for today's session. Today we are delighted to welcome Ms. Nitya Mittal, an accomplished researcher and biologist with over 4 years of experience in the biotechnology industry. Her work focuses on building high throughput machine learning ready assay systems using cutting edge tools such as iPSC derived models, CRISPR screens, and high content imaging. Nitya has a strong track record of translating complex cell biology into structured, scalable data pipelines, enabling next generation discovery platforms, and accelerating innovation in biomedical research. Her expertise lies at the intersection of advanced cell models, data science, and technology development, making her contribution especially valuable in today's rapidly evolving life sciences landscape. We are excited to have her with us today to share her insights and experiences. Please join me in welcoming Ms. Nitya Mittal. Over to you, ma'am.
All right. Just give me a second and I'll share my screen. All right, just making sure you can hear me okay.
Yes, ma'am.
All right. All right. Hi, everyone. Thank you so much for joining today for the seminar. And thank you so much, Swapnita, for that introduction. As she mentioned, I've worked at the interface of stem cell biology and data science. Over the past few years, I've built some disease in a dish sort of models in using cell villages, mostly with iPSCs, and I have scaled them for experiments ranging from CRISPR screens to imaging pipelines. Now, I know that some of you may be students or early career researchers. I myself am an early career researcher. And sometimes we don't always have access to, million dollar microscopes or large scale facilities. And I think that's okay because, as we are growing, this is just the first step in the journey. And I have designed this session today to be very tool agnostic and resource friendly so that you can take some practical lessons that you can apply anywhere. And so basically what we'll cover today is a journey from hypothesis to readout and how to take a biological question, design the right in vitro model, generate data that's not only meaningful but also can be useful for future computational approaches like now AI. So maybe think of this as a small starter roadmap. And we'll start with what makes a good disease model, and then we'll move on to how to design backwards from what your phenotype would be, and then look at some trade offs between realism, throughput, and cost. And then at the end, we will focus on ending with how clean data practices today, sort of prepare you for an AI driven biology for tomorrow. And I think for me, here the promise is simple. By the end, I want to make sure that you know how to design a minimal but meaningful experiment, one that you and your collaborators and maybe even future algorithms can trust. And I always say that controls are cheaper than regrets. So, keeping that sort of in mind, let's dive in. Um, we will go from exploring question to data, designing the model, measuring what matters. Data done right is basically data hygiene practices and such. And looking ahead, obviously, testing our AI readiness. Um, I'm just going to skip all of this part, but just to mention few important things here. I love trekking, hiking, dancing, and planning travel. Planning being the key word here. I think the actual travel part is something that I enjoy a little less compared to how much I enjoyed just planning travel. So let's dive right into it. What is a disease model? In my opinion, it's basically a mini version of a disease, but in a lab. And so, since we are focusing today a lot on in vitro disease models, I think it's important to keep in mind that, science is careful guessing, but with receipts. You are thinking of the disease model sort of as a stand-in actor, not the whole movie, but just enough to like rehearse a scene. And we all need disease models in order to conduct research. And it's for simple reasons, right? Like you can't have humans in the lab every single time to perform every single one of your experiment. It can't be performed in vivo inside humans, inside living things. And so we need many versions of disease in our lab. What disease models really help us do is connect our biology to a specific mechanism that you're trying to explore or study in that disease, and translate that sort of into a therapy, cell therapy, or immune therapies, or different kinds of therapies. It also provides us with reproducible, testable conditions, meaning that you should be able to perform your experiment over and over and repeatably again to get cleaner and cleaner data. With humans, imagine if you had three different patients and three of them would have completely different biological background, and you would test something like this and you would always get varied results, right? But there would be so many other factors in their life affecting your end of results. But when you make a mini version of disease in the lab using maybe say their cells, you're able to do a lot more standardization as well, and think about what other internal cellular biology might affect your final result. You can also allow some early screening before animal or clinical trials, making sure that everything that you're doing is safe. You can basically study what the toxicity index and are like when you're doing it in cells in vitro, and then eventually in animal models start at mice, and then going into primates, so monkeys, and then usually going in phase 1 into humans. So you can make sure that all the compliance stuff, as well as making sure that the toxicity index and everything is staying at a certain level before it gets into a human. So to summarize, basically you could say a disease model is an experimental system, whether it can be in the cellular areas, it is also in the animal and or the computational, that reproduces specific aspects of human disease, which enable us to study mechanism and obviously potential therapies. And okay. And so, which brings us, how do you design a disease model? Like, now you have this question, say, I want to solve ALS or I want to solve Alzheimer's, but what does that disease model, a disease model for Alzheimer's, sort of look like in the lab, and how do you even go about designing one? Well, I think I have always taken this approach of designing backwards. Usually, you are taught that, do literature review, do this, do that, and then, come figure out, make an experiment, think about where you want to start and, how you want to go about it, what would be your methods and materials used, etc. And then, think about what your analysis would be and how do you want to present your results, right? That's usually the pattern. When we think about designing backwards, you start with the outcome, right? Like what do you want your outcome to be? You are starting with the end. So you're basically thinking the question first. And if you think about it in your standard way versus designing backward, there are two questions on both ends. When you're doing designing forward, you are starting with a goal, but with designing backwards, you essentially start with the question. Example that if I want to know whether stress kills neurons, my phenotype becomes survival, right? Everything else, the cell choice, whether to do a co-culture, make a cell village, or to introduce perturbation, should serve that phenotype, right? This keeps our experiments focused, efficient, and also reproducible. Basically, you were saying, the very first point, decide the outcome. What phenotype would prove or disapprove your hypothesis? So, another exam could be neuron survival under oxidative stress, which is basically the phenotype becomes how many number of live cells do I have? What is my readout? Live versus dead stain. How do I, once I do that, what is going to be the measurable of that? That is your cell count. So your second pointer, which is define the measurement. Which assay or readout can capture the phenotype that I want quantitatively, right? And so the other example here could be TDP43 mislocalization in ALS in motor neurons. Here, my phenotype is nuclear versus cytoplasmic localization. People who have ALS will have cells where TDP43 has mislocalized from nucleus to the cytoplasm of the cell. And so the readout here, how do you see whether TDP43 has a protein has mislocalized from the nucleus into the cytoplasm? You can stain the cells and look at immunofluorescence intensity ratios. And so that's your measurement here, right? That's your readout here. Say you have done that, and you have some cells that are from donors and patients who have ALS, and then you have some from patients who don't have ALS. And in this case, you can just run an experiment. You can just run the measurement. You can just try to go with the readout and get your answer. But say you are a lab that only has iPSC cells and you don't have ALS cells, but you want to study ALS at some point and you have to start somewhere. What do you do then? That is when you select a perturbation that needs to be introduced into the cell. Now, that can be genetic, chemical, environmental, either, right? And so perturbations are basically edits, right? That you're introducing into an environment. So you, in this case, would introduce a genetic change in order to make your disease model. You will culture a healthy iPSC. You will get that from an iPSC day 0 to say a motor neuron day 10. And in before you do this day 0 to day 10, you will insert a construct that will give this cell cryptic neuron or ALS like morphology, right? So in that case, you have selected a genetic perturbation that will drive the phenotype, which is TDP43 mislocalization. Because this cell now has ALS construct, it will express mislocalized TDP43 from the nucleus into the cytoplasm, correct? Now you have, do you know in a way that what your outcome is supposed to be. You have defined a measurement, you have selected what perturbation to introduce. This could have been chemical or environmental too. Environmental could be like an oxidative stress situation, which we did for neuron survival in example. And chemical is, you could introduce a treatment onto it which leads to activation or inhibition of certain pathways within the cell. And so once you have done with your perturbations, you have also technically picked a model. If you realize, you have chosen, say, iPSC cells in this case. But if you hadn't chosen any cells and you knew that you were going to do genetic perturbation, you would realize that you are technically picking a cell type that can be edited, right? So that's your model right there. Which cell have you picked and which cell would be enough include to show your perturbation? And then you would add controls. I think controls is definitely the biggest part, and I emphasize this a lot. It basically would help you compare the good from the bad, or if there has been any changes. Not all of your cells can be treated with a drug treatment, so you definitely need controls to ensure that you're getting not just noise, but also you're reflecting, showing biology that is happening in your dish. Now coming to the three-way trade-off. This is something that I've adapted from a project management, a triple constraint triangle, essentially. So the way they do is they do it for fast, cost, and quality. And so I try to adapt this to a lab setting where realism, throughput, and cost sort of sits at the three corners, if you see. And in a way where it sits in these three corners, you would realize that when we say realism, we are looking for high insight. How closely can the model mimic the actual human disease biology? When you're thinking throughput, you're thinking how many conditions you can test quickly. And when you're thinking cost, obviously you're thinking money, time, and expertise that would be required in order to make sure that your disease model is a success. So, what are the orange bits? We have studied what the orange bits are. Now, what are the green bits in this triangle? The green bits are when you combine realism plus throughput, you can make a model that is expensive. It will cost you. You can optimize two, but it's very hard to optimize all three parts of this triangle. You have to choose based on your research question. So when you combine realism plus throughput, in this case, realism would be iPSC derived, motor neurons, and also using simple assays, but it could still be expensive. But when you combine throughput and cost, you might often end up with something that might not be highly relevant, and your disease model might not give you great phenotype. But when you combine realism and cost, at that point, you are building something which takes a little bit time because you're trying to go low expense, but you're still trying to make sure that you're producing the best quality stuff. So it's going to take you a lot more time to get results and deliver results, right? Um, when you think about throughput, you're thinking immortalized lines. They're great for screening, but less physiologically relevant. In case of cost, it's obviously simpler system will always save you resources, but you miss out on subtle disease specific mechanisms. So if you really have to think about it, you could think that if you need deep mechanistic insight, you have to prioritize realism, even if the throughput is low. If you need broad screening, you need to pick throughput and cost efficiency, but accept the lower biological relevance. And if you want clinically relevant insight at scale, which a lot of therapeutic companies do, right, to actually put a drug out there in the market, you need to prepare for high cost, rare outside big labs and consortias. You have to purchase data from other, online publicly available resources. You have to invest a lot of money should your preclinical studies get approved. You also have to prepare for scale because you won't be doing it once, you're going to be doing it again and again. And that requires money as well. And so I think here the question to ask when you are, say, designing a research, right, an experiment, what's the minimum model that will answer your question credibly with the resources that you have? Right. So you have to think about this triangle and then you have to see what you are trying to do in the lab. Where does it sit? What combination of these three things? Again, you can optimize two, rarely all three. So you have to choose based on your research question. So once you have designed a disease model, you have to think about if you're at a point where you're trying to think about, okay, what assays can I include in my experiment? You can think about this triangle and it sort of helps you be like a guiding light. Okay, this assay is possible or this assay is not possible. Now we come to choosing your cells. Naturally, when you're doing these things, you have to think about how are you going to choose your cells. The cell model that you choose is going to define how realistic your results are, how much can you actually test, and how far your findings might actually go. And in this case, I've included some images from under the microscope just to show you what some of these cells look like. If you have worked with some of these cells, amazing. I have not worked with all of these cells. I have worked with a few of these. I would use an immortalized line in my experiment. If I wanted to do something quick, cheap, fast, dirty, and have a lot of growth, it's great for learning techniques and some high throughput screens, but they don't have a lot of disease relevance. I think a lot of places try to start from these cells, but eventually you have to move on to iPSCs or primaries to get higher disease relevance as well. And if you think about immortalized cells, most immortalized cells would be flat, fibroblast-like, or will have some sort of epithelial morphology. If you zoom into this image right here, you'd be able to see that they're mostly flat and fibroblast-like. I would use primary cells in the case where I want more tissue-specific phenotypes. This type of cells are used often in kidney studies or liver studies as well. It's great because they have limited lifespan, but you can do a lot of quick assays, a lot of time point assays. They have variable quality, that is true. It depends from vial to vial. They are definitely harder to scale because they don't divide very well after the primary stage. And they are definitely more delicate, irregular in shape, and when it comes to primary neurons, the neurites can often break. So in that case, you have to handle them with care. And so they're basically like babies of all the cell types where you have to take a little bit more care. If I wanted to do something that was highly disease relevant, I would go with iPSCs. But like we studied in the previous concept, the three-way trade-off, you can't have everything. And hence, so iPSC is expensive, it is more time intensive, and it requires a certain kind of expertise to culture. iPSCs grow mostly in tight, compact colonies. And you can see in this image here that they have mostly clear edges and smooth borders. They are more patient specific or can be more patient specific, especially since induced iPSCs are reprogrammed. You can reprogram somatic cells into iPSCs. And you can also differentiate iPSCs into many cell types. That is exactly what takes a lot more time, actually. In differentiating them into different cell types, you have to come up usually with a protocol, unless you're buying it from a vendor who's going to give you all the media that, okay, do this for the next 10 days and you'll have this cell type. And if you're going that route, usually it's expensive. But if you wanted to, say you had the resources and you want to get even more close to understanding some sort of disease morphology, you could go down the organoid or the 3D culture route. Organoids are spherical like this in the image that you see down here. They are sort of, cyst-like, budding. I often like to compare them to almost very few organoids. If you culture them enough, you'd see they're almost like yeast budding structures, but they eventually become all spherical. And they actually capture tissue architecture in the best of ways. Great for studying complex phenotypes. They are even more resource heavy and very less scalable. This comes from my own personal experience as well. Organoids take a while to get there, and iPSCs become, the first choice if you are going to, if anybody who's considering organoids always goes iPSC colonies as their first choice. So, the takeaway from here in choosing your own cells is the right cell model will have a good balance of realism resources, but at the end would always answer your research question. So it does not have to be a balance of all three, but mostly realism, resources, and your research question. Thinking about perturbations. Perturbations are basically, how we stress test a disease model. We want to make sure that it works. We want to make sure, when we poke the system, it reveals a phenotype. And make sure that, anything that we introduce, if it has an effect, we are able to capture it in a measurable way. So I would say the different type of perturbations, let's go over them one by one. There are only three main, but I include controls as non-negotiables in this slide for a reason. So let's start with genetic, right? So genetic perturbation is like hard coding the disease into the cell. You can knock in a gene, you can knock out a gene, you can knock down a gene. That can be done via CRISPR. You can also use siRNA or shRNA treatments, all of which would help you mimic the disease mutation or silence or activate certain genes. And in terms of chemical dosing them with a stressor or a drug essentially will also inhibit or activate pathways. And you can do that by using small molecules, toxins, or drugs. They're actually pretty easy to apply. It's essentially just, adding certain concentrations. They are dose dependent. So the readout becomes dose dependent curves, and you would see that different doses, the cell survival rate, the cell toxicity rate. And usually these are reversible depending on what the concentration of the drugs, toxins, or the small molecule was. In terms of environmental stress, it can be hypoxia, it can also be oxidative stress, it can be nutrient deprivation by altering the media, it can also be temperature shifts. Sometimes when you're running assays, you have to change temperatures between like 37 to 42 degrees. In my experience, I've run some assays that require multiple steps, and your cells go from 37 to 42 to 41, and sometimes as low as 30 degrees Celsius as well. So, what this does is it basically mimics external stress conditions that might be relevant to the disease. And coming to the last one, which is controls. Controls are basically telling us, is the effect real or is it just background noise? You definitely need a negative and a positive, right? Think of something like a simple assay like flow cytometry. You are running an assay that has iPSC cells, which express certain markers that say that, okay, if this marker is present and this marker is present, it's going to be an iPSC cell. But then you do a motor neuron, right? And so the motor neuron might have two separate markers, right? And so now your control becomes iPSC. Say you were differentiating this iPNC into a motor neuron, you want to see how well it was differentiated. So your control here is just a pure 100% iPSC versus a day 3 motor neuron versus a day 10 motor neuron. And all three of these different cell types at this point are going to express different markers. And now if you didn't have an iPSC and you just had a day 3 and day 10, you might not be able to say like which one has been differentiated more if the time difference was not a lot, right? Between day 3 and day 10, you'll have to see, okay, which one has a more mature neuron marker versus which one has a more less mature neuron marker. Becomes a little bit confusing, and then you don't really know what it compares against with the iPSCs. So controls always important. In terms of environmental, it's making their world a bit harsher, making the world of cells a little bit harsher, less oxygen, less food, more stress. Chemical is like dosing them with a stressor or a drug, and then genetic is like hard coding the disease inside the cells. Now, coming down to affordable readouts. So far, we have, perturbed ourselves as well. We have made sure that they are getting some kind of stress. But after all is done, how do you measure what is happening inside the cells? And how do you measure it without breaking the bank? A lot of times, readouts are the most expensive part of the experiment. And again, this is, throwing out there the most basic readouts that you can get without any fancy equipment with some basic equipments in the lab. So I would say we have to start with measuring what matters, right? And the very first thing is morphology. You can use a bright field or a phase contrast microscope, a simple microscope that will tell you what the cell shape, size, and clustering is like. And your first line of thought is something happening? You culture the cells, you look at them every day in the microscope from like Monday, Tuesday, Wednesday, Friday, and you're noting down differences across each of the days. And that sort of tells you if something is happening at all. And remember, affordable does not mean trivial. It basically means smart use of what is accessible. And starting with morphology and viability, you would be surprised how much biology is visible under a simple scope. The next thing I go to is viability assays. And viability assays have been used as, like holy grail in the last few decades of research as well. And it's very simple. You can use just your regular stain, trypan blue, live dead stains, and there are a lot more different viability assays that can be used apart from just your cell count, which are also pretty easy to use. You're basically trying to pick one or two quantifiable readout and do them well without trying to chase fancy multiomics. And some functional assays, calcium dyes for activities in neurons is pretty common. You don't even have to develop the whole assays. you can see there are some papers out there who tells that will tell you, if you're doing this correctly or not, depending on, what your research questions goal is. I know there is MTT for the metabolism assays, and this is usually also done for cancer cells, if I'm not wrong. Um, when you do a drug treatment on cancer cell, you can test it out how the cells are performing using the MTT assays to check the metabolic activity in the cells. Alternatively, for oxidative stress on neurons, which is used across multiple neuronal disorders, you can count the percentage of shrunken versus normal cells, and that can be done just using a bright field microscope. On the right hand side, the image basically is, the different kinds of things that you could target, and the different kind of assays, the result of those assays could be. Like, if you go into depths of every single one of these, you see that there are multiple easy ways to do some quick assays to learn a lot more about your experiment. Immunostaining is still considered pretty low plex. And there are a couple of protein markers that are very easily available as well. And it does not require super high end multiplexing either. And so you do not need a very fancy microscope to do some immunostaining and study it under the scope. Plate maps. This is more like a plate maps and randomization section. And you would see that I have three different types of plate maps on here. The reason I included plate maps as a section of disease modeling is, once you have picked your readout, now you need to design the layout so that your data is also trustworthy. So in the very first image, you'd see that it's a 96 well plate layout and it has some numbers in it. These numbers are likely, this is an image of internet, so these numbers are likely chemical dosage concentrations, like how much is being put in there and at at what value, and you can see the gradient whether it is high or low. You want to make sure that mapping and randomization is reducing hidden bias before or your confounders sort of start sneaking in. I also want to drag your attention to the second image. This is a confidential image. This is an image that was part of my own experiment. It is, again, a very simple but effective experiment. We have two things that are happening here. We are using day 10 motor neurons and day 30 motor neurons. Both of them have two replicates. One plate is with astrocytes, one plate is without astrocytes. The base cell here is motor neurons, and I am trying to test my control in both the cases, which is DMSO, and then I'm doing drug treatment, which is a chemical stressor of bortezomib and puromycin. And so later I can study their drug dosage curves, and I can also study how it has affected my neurons under the microscope. On the third image, it's a regular image where you can see that, if I had to set up the most basic experiment in a 96 well, how would that be? I would have some control cells and I would have some treated cells. And you would see that in this image, we have left out the edges of the plate. And I'll tell you why we are doing that. So the basic plan for doing some plate mapping is, one, you're drawing or printing a map and you're assigning the wells very clearly, and you're going to try and avoid edge effects because there is drying that happens on the edge effects. Usually the media will evaporate faster from the edges, so you try to leave out. Even the first A row and the H row ideally should have been left out in all of these images if you're trying to do like a good plate map. And then, obviously coming to the next part, outer wells dry faster. This is going to distort your results and it is going to interrupt the quality of your data as well. So you have to leave out the edges, but you also have to use randomization. Now, in this last image, you see all the control cells and all the treated cells are together separated very distinctly. But if you see the first plate, it is highly randomized. So if I had to choose a good data set, I would choose the first plate because it is randomized. You have to spread conditions across the plate or rotate assignments across the replicates. So that again points us back to the first plate. The first plate is a great example of a good plate map, not in terms of outer wells drying faster, but in terms that it has great randomization to it. And there are softwares that will, where you can just plug in quantities that, okay, I have these many cell types, these many conditions in a randomized way, put it on my plate map. It is a lot more work to do manually, but this gives you the best results. It makes sure that your results are reflecting biology and not bias. You have to make sure one other part that you're focusing on replicates. So say you have cells from patient A, B, C, you need to make sure that you have minimum of 3 replicates. So minimum 3 wells per patient. You will always have more than that if you're working with 96 well plates. But say you are working with a 6 well plate, like the second case, you need to make sure that you have at least 3 replicates. Okay. So that basically is going to ensure biology and not layout bias. Randomization does not mean chaos. It means designing so that your treatment and control conditions are exposed to the same plate environment, which is why, again, coming back to that point of leaving the edges blank. All right, coming to watching confounders. So confounders is like background noise from your, when you wear headphones, if you have like headphones like mine, which are cheap, uh, if you wear it and you hear background noise, that's a confounder. If it's too loud, you can't hear the music clearly, right? So confounders are basically like hidden troublemakers. They will sneak in and will spoil your data set essentially. It's a hidden factor that's going to change your results, not by a lot sometimes, but they change your results without you noticing them. It will make you think that a concentration of some drug caused the effect when it absolutely did not, right? So in cell biology, density and passages are usually classic culprits of being confounders. They change phenotypes. Even small things like, how long the media sat in the fridge can change your result. And usually good science is spotting confounders early and controlling them. So in terms of cell culture, some common confounders are cell density, passage number, media freshness, even operator differences. In my last few experiences, we have tried to slowly automate things, even whether it's the smallest, the difference between just changing media using, say, a wire flow, Integra wire flow, versus doing it by hand. It matters which person does it. So on a team, if there are 2 people, operator 1 does it a certain way, operator 2 does it a certain way. And if that changes your result, that is a confounder, which is why different people, different results, confounder. And sometimes the daytime effects also, change it. Your how your cells were looking on Friday versus, when you came back on Monday, if they're looking a bit different, that can also be a confounder. And why do confounders matter? Because they blur the signal from your real experiment. They can waste weeks and months of effort if we are not tracking them. So good science is spotting confounders early and controlling them. Coming to data hygiene. And so, we have managed to control our confounders, but our results only matter if we can manage our data well as well. So data hygiene focuses on making sure that everything that you're producing is labeled, organized, and structured so it is interpretable months or even years later. And this comes back to your age old saying that our lab managers, that our managers say, our PIs say, that make sure that everything is tracked and done well. So if tomorrow so I had to come in the lab and perform the experiment, I could read your lab notebook and do it. And so Obviously it matters because bad naming and messy folders is equal to wasted time, lost results, unusable for analysis. Pretty true. And for being someone who has generated lots and lots of experimental data till date, I can say the worst pain it really is when you come back to a folder, even say on a microscope, and the file says day one, no cell type, no time point, just day one. And then you have to look at the file and you start thinking about, okay, when did I take this image and when can I use it? Is it even good image to put in my slide decks? You have to start thinking about all that. But if you ever want your experiments at the end in the age of AI to feed into an algorithm, the very first step is not getting a fancy algorithm. It's just a clean, tidy data set. And so, some simple habits that you can do is use clear, consistent file names, come up with your own format, what works for you. I always include an experiment date, a version if I'm going to be performing multiple, and a condition if there is one. And I sometimes doing this, having this system sort of makes it very long, to very long file name, but it also helps you keep track. Then you can have a simple metadata sheet, which would have your cell type, passage, density, treatment operator. I always maintain an Excel sheet or a Google sheet on the back end to make sure that every experiment that I'm doing is recorded. Everything that I am going to be performing in the lab is getting recorded, whether it is cell counts or anything that's happening. So it's basically a timeline for your experiment. If you're somebody more of like a Google Docs or just Microsoft Word person, I would still write down what happened or what you did on the lab in that day, just so you have something to come back to. And store raw and processed data separately. I know that it's easier to just dump everything that you did on one day into one folder, but go back and organize it always. Like, if you had something that was like, a heavy file, like a 2 GB or 2 TB even file, which is raw, and then you are going to analyze this later, or say you have analyzed it, don't put it back in the same folder. Keep your raw data and your process data separate always. And then obviously version your protocols and plate maps. Don't overwrite them. Even if you think that this is the final thing that I'm going to be using, don't overwrite them. Just version it. This is like my version 2, version 3. No using like final or final file. Just put a version to it. The long game is clean structured data today is equals to data sets tomorrow that can be used for machine learning and AI models. Remember, future you is your most important collaborator. Label files like you respect them. You are probably the only person who's going to come back and use them as well. So, coming to basic analysis and quality control, and we'll be wrapping up soon after this slide. So you can, ask any questions if you have. Basic analysis and quality control includes, going from raw data to trustworthy results, of course. So far, we have talked about building models, avoiding confounders, and keeping data tidy. But all of that only matters if the analysis step is honest, reproducible. If not, what's even the use, right? And so here I want to say that basic QC just means looking at your raw points, looking for patterns, but also looking for noise. In most cases, if your treatment effect disappears when you show error bars, you don't really have a story yet. You need to revisit everything that you are doing and see what happened there. This part, analysis and quality control, is the bridge to AI readiness. An algorithm can only learn from data if it reflects true biology, not hidden mistakes. If you want to go deeper into, practical QC statistics or experimental design, I am sure that Biopractify runs really great courses where they teach you, more into bioinformatics beyond obviously what we are covering in this webinar. But yeah, I would say these 5 pointers are basically your backbone for basic analysis in QC. predefining analytical endpoints, visualizing raw data early, quantifying effect size plus uncertainty, and detecting outliers systematically. And obviously, at any point, anything, not just your analysis and QC, you have to make sure that everything is reproducible. So document, document, document. Next pointer and our last for today is AI readiness. So I wanted to include this obviously because we are moving in that direction and I am really enthusiastic and I do work at the intersection of AI x biology and I want to continue working there. But there are some things to remember when you are moving towards it or into it. AI can't fix messy biology, it only exposes it faster. So here it's not about just data hygiene, right? Data hygiene is the first step. Data hygiene is about not losing your work. AI readiness, on the other hand, is about making your work useful for pattern recognition because that's what most algorithms do. If your phenotype is only described in words or pretty images, AI can't use it. Like, just immunocytochemistry, hard for AI to use. It can still use, it can still run segmentation, but you do need numbers. You do need intensity ratios, counts, and neurite lens, etc. You need a model to be trained well because a model trained on sloppy labels will only learn your sloppiness. So starting small is okay, but keeping it structured is important. You don't need fancy tools. Just well labeled spreadsheets are a very big foundation of AI biology. Start thinking in the way of an algorithm. What numerical features would capture your phenotype, right? Counts, ratios, intensities, textures, what kind of segmentation do you need to run? Nucleus, cytoplasm, more cell organelles, etc. And then you have to think about designing for generalization. So keeping training and validation as separate, avoiding overlap for donors and conditions. And in cell villages, that's a very good possibility. So if you ever go down the route of cell villages, you have to start thinking how to keep those things separate. You have to standardize your formats. So I mean, a lot of companies do this. You are supposed to work, in a certain way. Files that are going to train models are going to be using consistent unit scales, files type, etc., across different experiments as well. And second last is to plan for scale. You are going to always collect data, but you have to collect it in a way that it can grow into larger data sets later. I think planning for scale is a lot more reflection of, also planning for reproducibility. And lastly, obviously, ethics matter. Transparency in data preparation is as critical for AI biology as biology itself. So, making sure that obviously we all adhering to the moral standards and ethical standards. All right. I know I don't have a lot of time, but any questions, any curiosities?
Participants, you can turn on your mic and ask the questions. Also, you can write it in the chat box. Any questions, you can write it in the chat box. Hello, ma'am. I don't think we have any questions at this point.
No worries. I can go ahead and stop sharing my screen though.